I'm a professor at King's in the Cardiovascular Division in the British Heart Foundation Centre for Research Excellence. I've been at King's probably over 30 years. I'm a research active scientist interested primarily in how vascular cells fail in disease. Recently, or probably 5-6 years ago, we began asking the question whether cells in culture are really seeing the correct physiological oxygen concentration. And the answer is absolutely not. Because the Baker-Ruskinn equipment is instrumental for doing the kind of research that we do, we need to have an environment in which cells are isolated, subcultured, treated and harvested, all under a defined oxygen level. The in vivo product allows us to take endothelial cells and smooth muscle cells and cardiac myocytes from an environment that is non-physiological, hyperoxic, into an environment that is physiological, to manipulate the cells in a closed environment, regulated by defined oxygen levels, and then to begin to probe these cells with stimulators, with fluid shear stress, with mechanotransduction, and to identify signaling pathways inside the cells at defined intracellular oxygen levels. I think it's the enthusiasm and interest in science from colleagues in Baker-Ruskinn that have led me to stay with them. I think that Baker have listened, and I think that Baker-Ruskinn have clearly taken on board suggestions, at least from our group, to facilitate the type of research we undertake. And this is the focus of the future for us.
And we can put our 3D technology inside these environmental control chambers to then enable us to enhance cellular differentiation processes. And we're particularly interested in the formation of human skin models. Maintain stability and consistency of the environment over time. So have a long tradition of working with scientists and have excellent interactions with research leaders in the field. In terms of controlling oxygen and carbon dioxide temperature within an environmental chamber, Baker-Ruskinn certainly are leaders in the field. They've developed technologies that enable you to have fine control and monitor the conditions and set the conditions that you require as a scientist to enable you to more closely mimic the growth conditions found in the body.
Hi, my name is Tom Keeley. I'm a PhD student in the lab with Professor Giovanni Mann at King's College London. So I use it almost every day, routinely. What we think is fantastic about it is the fact that because it's a closed system and it's easily accessible, you can perform everything you can do on the bench inside the chamber, and that allows our research to really progress. So I feel that Baker-Ruskinn is a pioneer in this field of both industry and research, and they're really paving the way, especially with the release of their new in vivo models, to facilitate the introduction of this, what we term closed culture in defined oxygen levels, to make it more accessible for research throughout the world. So I believe through the use of the Baker-Ruskinn products, we are better replicating these normal cellular behaviour by recapitulating their in vivo environment in vitro. The ease of use which we experience with the Baker-Ruskinn products allows us to introduce this new technology. The results we are gaining from the last few years of research really are painting the picture that the cells grown under physiologically relevant conditions are better representative of the cells that are growing in your body. My only experience is doing it under a physiologically relevant environment, and I can foresee in the not too distant future abandoning room air culture completely and performing all your culture under physiological conditions.